Hello, my name is Thomas Hoang, a researcher at UT Health, McGovern Medical School, in the Department of Pediatrics Division of Gastroenterology, led by Doctors Mark Rhodes and Yu-Ying Liu. On behalf of our group in Houston, Texas, it is an honor to share this work with you that was recently published in the American Journal of Physiology, Gastrointestinal and Liver Physiology. Necrotizing enterocolitis, or NEC, is one of the most devastating neonatal diseases. The incidence of NEC in preterm infants around the globe varies from 2 to 7% in neonatal intensive care units. Recent studies that have investigated the pathology of NEC have shown that it is promoted by uninhibited activation of toll-like receptor 4 in response to excessive proliferation and translocation of pathogenic microbes. This leads to an increased inflammatory response. Probiotic supplementation has correlated significantly with the decreased risk of developing NEC in very low birth weight infants. Our group has studied and published on Lactobacillus reuteri DSM 17938, which was originally isolated from a Peruvian mother's breast milk. This gram-positive strain has shown to inhibit pathogen growth, modulate the immune system, and have strong anti-inflammatory effects. We postulated whether L. reuteri 17938 provides protection through recognition of a toll-like receptor. Unlike toll receptor 4, toll receptor 2 recognizes cell wall components of gram-positive bacteria. Previous studies indicate that toll receptor 2 signaling enhances barrier function in the intestines. Ligation with TLR2 using synthetic agonists have shown to suppress mucosal inflammation and intestinal epithelial cell apoptosis. In our study, we examined the effects of TLR2 deficiency on the protective effect of L. reuteri 17938 during the height of experimental necrotizing enterocolitis in a well-established neonatal mouse model. The animal model of NEC we use includes formula feeding of infant mouse pups with subsequent exposure to cold stress and hypoxia. A combination of asphyxia, formula feeding, and a lack of mother's breast milk play a key role in the pathogenesis that is shown to mimic human NEC as evidenced by histology. Control groups of wild-type and toll-like receptor 2 knockout newborns were breastfed and housed with their dams throughout the 4 days of NEC procedures. Experimental NEC groups were weaned early and housed in an incubator 12 hours before oral gastric feeding with a specific formula 4 times a day, daily, over 4 days. Wild-type and toll-like receptor 2 knockout pups were formula fed with or without L. reuteri. Including experimental control groups, there were 6 groups total. Pups were euthanized on day 5 after NEC procedures for tissue collection. The terminal 5 centimeters of the small intestines or ileum was collected for several purposes. A portion was used for H&E staining for histological evaluation. Another was quickly frozen for processing tissue lysates to examine cytokines using ELISA assays, and another was immediately used to isolate lymphocytes to analyze by flow cytometry. This graph shows the incidence of NEC of each group. Each mouse of each group were given scores from 0 to 3, and a value greater than 2 represents NEC. After NEC induction, there was a dramatic increase in the incidence of NEC in both wild-type and toll-like receptor 2 knockout groups. The incidence of NEC in wild-type mice with L. reuteri supplemented formula significantly decreased. However, the incidence did not decrease in toll-like receptor 2 knockout animals, remaining at 75% incidence in our study. L. reuteri increased Tregs and tolerogenic dendritic cells after NEC, which are both non-significant effects in toll-like receptor 2 knockout mice. In our studies, the beneficial effects against NEC by L. reuteri required toll-like receptor 2 to activate the immune signaling by dendritic cells that prime T lymphocytes to promote tolerogenic outcomes. This was seen by the increase of regulatory factors and the reduction of inflammatory cytokines in effector T cells. The direct or indirect interactions of L. reuteri with immune cells, the possible effects of microbial composition, as well as the intracellular signaling pathways that contribute to homeostasis, warrant further exploration. In conclusion, these important findings shed more light on the regulatory and therapeutic activities of L. reuteri during experimental necrotizing enterocolitis.